functionality, which can lead to greater fat usage as well. So this is where astaxanthin comes in. Astaxanthin, I know it's kind of a hard word for some people to say, but it's derived from microalgae, and it's what's called a carotenoid. So that just means it's a red pigment. And it's what's found in salmon that gives it its color, crabs, a lot of like shrimp, that kind of stuff. And another carotenoid that you've probably heard of is beta carotene. So beta carotene has also been shown to work as an antioxidant, but the research suggests that astaxanthin is actually a better carotenoid, a better antioxidant than it is, and it has to do with the actual structure of astaxanthin. So what this figure is showing, so this is called the phospholipid bilayer. So this is what encompasses your cell. So this is kind of the outside part of your cell. And on the outside is polar, and the inside part is nonpolar. And because of the way the astaxanthin is, it's also polar on the ends, but not polar in the middle, or nonpolar in the middle. And because of that, it can actually fit perfectly in the phospholipid bilayer. So in doing this, it can protect stuff from coming into the cell and also keep stuff from leaving the cell as well. And I'll go into that into more depth. So astaxanthin has been shown to improve mitochondrial function, which can lead to decreased oxidative stress and increased fat oxidation. So not only can it improve the functionality, but research has shown that it can cause increases in what's called peroxisome proliferator activated receptor gamma coactivator 1 alpha, which PGC1 alpha is a shorter way to say that. PGC1 alpha has been shown to cause mitochondrial biogenesis, which is the production of mitochondria. So if you have more mitochondria, you have more places where you can actually have that fat and carb protein breakdown, so it's less taxing on all of them as a whole. And with more mitochondria, you also have more enzymes that come with the mitochondria, again, allowing it to be more functional, as well as increase your fat oxidation. And like I said earlier, if your mitochondria function better, you're less likely to have that leakage of the superoxide anion, which can cause oxidative stress. Astaxanthin can also act as an antioxidant alone. So I'm going to go back to the previous image here. So like I said, it can span across the phospholipid bilayer, and whenever oxidants try and leave the cell, it can actually catch the oxidant before it leaves to either take one of the unpaired electrons or to donate an electron to it so that it becomes more stable. So it can act as putting the cotton over those sticker burrs in the cell. It basically acts like a net. And research also shows that while most antioxidants can take care of one oxidant, so one antioxidant can put the cotton over one sticker burr, astaxanthin can actually take care of 19. So for every one molecule of astaxanthin, we can actually take care of 19 oxidants, which makes it work, or suggests that it works better than other antioxidants as well. And astaxanthin has also been shown to cause increases in antioxidant activity. So I'm going to go to this next slide here. So this is what I showed earlier. So SOD and glutathione were the antioxidants that I briefly talked about before. And research suggests that both of these can also be increased with the supplementation of astaxanthin. So not only can astaxanthin act as an antioxidant, but it increases other antioxidants that are already in the body as well. The purpose of this study then is twofold. We wanted to evaluate the impact astaxanthin has on postprandial oxidative stress, which is oxidative stress after eating, as well as substrate utilization during exercise. So that's the carbon fat oxidation rates during exercise. The methods for our study, so we had 13 participants that were all healthy males, 18 to 39 years old, no history of cardiovascular metabolic diseases or blood disorders. We asked them to not take any supplements that would impact the study, make sure that they weren't smoking, because smoking can cause oxidative stress, and minimize red wine and coffee consumption because those have been shown to cause increased antioxidants, as well as not participate in veganism because that can also have similar effects. Participants were recruited by word of mouth and flyers. The research design was a double-blind, randomized, placebo-controlled crossover study. That just means that the participants took both treatments. So each participant did the placebo and the astaxanthin treatment. And whenever they did it, so if they did placebo first, they did astaxanthin second, and some of them astaxanthin first, placebo second. And down here, you can see the demographics of the participants. The reason why it's broken up into two sides is because one of the participants in the postprandial oxidative stress portion was not in the fat max, and one of the people in the fat max was not in the postprandial oxidative stress. So that's why those demographics are slightly different, but they're still pretty similar. And the percent compliance just means how many days, basically what percentage of the days they actually took their supplement. So they were asked to take it once a day for 4 weeks. And for the postprandial oxidative stress people, they had 95% compliance, and the fat max had 92% compliance on average. So these were the unmarked pill bottles that I counted every single one of these. So we had 35 in each pill bottle, had to count them by hand, put them in the bottle, screw the top, and they were labeled A and B. So I didn't know which one was the actual astaxanthin and which one was the placebo. But as you can see, so these are the A's up close here. The B's are further back. They all looked the exact same, so there was no way to tell which one was which. Dr. McAllister was the one that had the code for that. So for the very beginning, we screened participants. So we got their height and weight, had them fill out consent forms. They had their first supplementation period, so I randomized whether they took the astaxanthin first or the placebo first. And for the astaxanthin, it was 6 milligrams a day. And then for the placebo, it was just safflower oil, which has not been shown to increase or change oxidation or oxidative stress in the body. And then at the end of the 4-week supplementation period, they had the milkshake testing day, which I'll go into more detail on that. And then they had the exercise testing day. There was a 1-week washout period, which the company that very graciously donated our supplements to us said that 1 week would be enough time for it to wash out of the system. And then we started the second 4-week supplementation period, again, ending with a milkshake and an exercise testing day. So for the milkshake day, or session 1, we had participants come in and drink a milkshake that was high in fat and high in carbohydrates. So right here, this line is showing our actual timeline. So whenever participants came in, they got a blood draw immediately. They went downstairs and they made a milkshake for the participant. So the recipes for the milkshake were based off their body weight. So if we had a participant that weighed, for example, 100 kilograms, they would have 100 grams of fat, roughly 100 grams of carbohydrates, and then about 25 grams of protein in the milkshake itself. Some of these people were a little bit taller. We had a guy that was about 120 kilograms. So his milkshake ended up being 64 ounces. So we had to have 2 32-ounce cups that he had to chug in about 15 minutes. But I think he actually finished in about 5. He took it as a challenge. But so it was based off of body weight. So it differed based off that. And the mean content is down here. So it was roughly 84 grams of carbs, 21 grams of fat, 26 grams of protein, which goes to roughly 1,169 kcals in one milkshake. And I'll come back to that later as well. So after they were done with the milkshake, we did another blood draw 2 hours after that. And then we did a blood draw 4 hours after the initial ingestion. So we had 3 total blood draws here. And what we did with the blood was analyze it for oxidative stress and lipid profile. So here are some of the pictures from data collection. So on the left side, we have the blood from the that we got in the vacutainers from whenever Dr. McAllister did the draw. And then we would analyze it, or we would treat it for whole blood, which is what these are for. And then we take the aliquot, or the plasma, or the clear part of the sample, and we put it in these cryovials to be stored in a negative 80-degree freezer. And here on the right side is just kind of us waiting for participants to come up, just having a great time for data collection, honestly. And here you can see the actual changes. So on the left side is plasma so plasma before they ate the high-fat meal. And then on the right side is after they had the milkshake. So you can see that it's clear on the left versus on the right, it's pretty cloudy. even the pictures don't do it justice, honestly, how cloudy the plasma actually got. But that's because of the amount of fat that's in your plasma. So a lot of people think of how food impacts you, you know, after a few days or months or years, but acutely it can cause so many changes in your blood. So here you can physically see the changes that are occurring in the blood, the amount of fat that is in your plasma. So for the blood analysis, we looked at hydrogen peroxide, TAG, which is just triglycerides or the fat in the plasma, AOPP, which is advanced oxidation protein products. AOPP is what we looked at to see how much damage was done to protein in the body. And MDA, or malondialdehyde, this is what we looked at to look at how much damage was done to fat cells instead. So protein damage, fat damage, hydrogen peroxide, I briefly talked about how it's an oxidant, and then TAG is the amount of fat in the bloodstream. And then whole blood was analyzed for glutathione, which I talked about earlier as the antioxidant that's naturally occurring in the body. So we'd start off blood analysis by taking out our samples. We would have them thaw. And this is one of the blood analysis days that probably took about 10 hours. So you can see up here screwing and unscrewing the tops of the cryovials so that we can get the samples ready, treating the samples, and getting everything to put into what these microplates. So these plates are actually about this big, and each well is about this big. So you can't really tell how precise and tedious the whole process was with this picture, because it makes it look a lot bigger than it was. But so each one of these has a sample in it. So these first ones are just standards, but these are samples from our actual participants, and this is what we put into the machine to actually look at how much of everything was in it. So for the session 2, for the fat max day, or the exercise testing day, this was the day after the participants came in for the milkshake day. They completed a graded exercise test on our cycloergometers. So each stage lasted 3 minutes. And over here, we have the resistance, or the watts, that was on the bike. So our first stage was 50 watts. And then at the end of the 3 minutes, we bumped it up to 80, and then 110, 140. And then after we hit 140, which was stage 4, we bumped it up by 50 watts each time. That way we could basically get the participant off the bike. Participants were going until they reached volitional exhaustion, which is where they can't pedal anymore. At the end of each stage, we use the last minute average to look at RER, which is respiratory exchange ratio. This is just comparing the amount of oxygen you breathe in to the amount of carbon dioxide that you're breathing out. So that's the ratio there. We looked at fat oxidation rates, so the amount of grams of fat you use per minute for energy, and carbohydrate oxidation rates, so the amount of grams of carbohydrates you use per minute for energy. So once they completed their fat max test, we had them cool down. The cool down was back down to 50 watts. We had them every 2 minutes, so 2 minutes in, 4, 6, and 8 minutes into the cool down, we took a finger prick so that we could look at lactate levels. And what lactate is a byproduct of carbohydrate metabolism in an anaerobic state. So that means if you have more lactate in the blood, you've been using more carbohydrates. So that's also another way that we could see whether you use more fat or carbohydrates with the astaxanthin supplementation. Also, for each of these days, 24 hours prior, we had them fill out a dietary recall. So we had them say everything that they ate the day before. And so for session 2, so the second time that they came in to do the testing, we asked them to try and eat the same foods that time too. So that way, as most of you know, whenever you eat something, it can affect how you feel the next day. So we tried to keep that as similar as possible for both treatments, whether it's the placebo or the astaxanthin. And these are the formulas that we use to calculate the oxidation rates. So for fat oxidation, this was the formula that we used. VO2 is just the volume of oxygen you breathe in. VCO2 is the amount of carbon dioxide that you're breathing out. And then this was the carbohydrate oxidation. So again, VCO2, breathe out VO2, breathing in oxygen. These were formulas that have been used by Asker Eukendrup, which is the person that made the FatMax protocol, as well as Dr. McAllister has used these oxidation rates formulas for previous research as well. For statistical analysis, we use a 2x4 treatment by stage repeated measures ANOVA for lactate, carbohydrate, and fat oxidation rates, as well as respiratory exchange ratio. We use a 2x3 treatment by time point, repeated measures ANOVA for hydrogen peroxide, glutathione, AOPP, MDA, and TAG, which were the blood markers from the milkshake day. And if we found a p-value to be significant, so less than 0.05, we use a Fisher's least significant difference post hoc to compare the means. So now for the results of the study, the fun part. So this graph here is showing TAG levels. So that's the amount of fat that was in the plasma. We have pre, 2 hours post, and 4 hours post. So we didn't find an interaction or main effect for treatment, but there was a main effect for time that was significant. So p-value is less than 0.01. So here we have 2 and 4 hours post were significantly higher than the pre-levels. For glutathione, so still the same time points, this was the antioxidant that naturally occurs in the body. We found that there was no interaction found, no main effect for time, so no change in time, but there was almost a significant increase with astaxanthin treatment. So while it was not significant, which would be a p of 0.05, it did approach significance, so we got to 0.06. For hydrogen peroxide, which was the oxidant that we measured, we found an interaction with a p-value of less than 0.01. So that means that this right here, so the thick line is the placebo, this is the astaxanthin down here. This change right here, so this increase in hydrogen peroxide was significantly higher than this increase in hydrogen peroxide. So you can see astaxanthin had lower amounts of hydrogen peroxide. Next, we have AOPP, which is what we measured to look at the amount of damage done to protein. We have the stats down here again, and we found another main effect for time, where 2 and 4 hours post were significantly higher than the pre-levels that we measured. And then for MDA, again, this is the damage done to the fat cells instead of the protein. We found also a main effect for time, 2 to 4 hours post ingestion. So 2 and 4 hours were significantly higher than the pre-levels. And again, here, the thick one is placebo, and then the dotted line is the astaxanthin. Here now we're switching to the FatMax testing day. So this first one is carbohydrate oxidation. We had a main effect for stage. So this is stages 1, 2, 3, and 4. We had significant increases at each stage in carbohydrate oxidation. And then we had the exact opposite for fat oxidation. So as we went stage by stage, fat oxidation significantly decreased. So we had a main effect for stage there, which is what you would expect based off of the previous research I talked about. As we increase intensity, carbohydrate usage goes up. As we increase intensity, fat oxidation goes down. Respiratory exchange ratio. So again, this is comparing the amount of oxygen you breathe in, carbon dioxide you breathe out. We found, again, a significant increase at each stage. So each stage got significantly higher, which suggests that there's greater amounts of carbohydrates used during the test as we increase intensity. So lactate, as I mentioned, immediately after the exercise test, we had them cool down for 10 minutes. Every 2 minutes, we took a lactate reading. So this is 2 minutes after, 4 minutes, 6, and 8 minutes after. 2 to 4 did not have any significant differences between them. 6 and 8 also didn't have any differences between them. But 2 and 4 were significantly higher than the 6 and 8 minute marks. So although there was no differences between these two and these two, there was that significant difference between 2 and 4 to 6 and 8 minutes post. So that, again, so lactate suggests that there's greater carbohydrate usage. So the greater lactate, the more carbohydrates you're utilizing as well. So interpretation, what does this all mean? So with postprandial oxidative stress with fat, MDA, AOPP, and glutathione, we did not see changes between treatments, but we did see changes over times. So this does suggest that our milkshake was successful in inducing oxidative stress in even healthy individuals. So the healthy individuals started off with healthy levels of all of these, but we were able to induce unhealthy levels, essentially, besides with the glutathione. So we didn't get any changes really with glutathione. So why didn't we see any differences between the treatments? Why didn't the astaxanthin impact it the way that we thought it could have potentially changed it? One thing is that we could have potentially not measured enough markers. So there are a lot of other markers of oxidative stress, other antioxidants that we could have looked at but didn't look at. Part of it is the amount of work that we still did was a lot of markers, and a lot of the research that was previously done specifically on postprandial oxidative stress, that's the markers that we looked at. And additionally, adding more markers also makes it cost a lot more money. Dr. McAllister was gracious enough to fully fund all of this, and it was already multiple tons of dollars analyzing the blood for the analytes that we looked at. And also the small postprandial window, we only looked 4 hours post milkshake consumption, versus some studies do look at 6 hours post milkshake ingestion or post high fat meal ingestion. The reason why we didn't look further is, again, that would add another blood marker or another time point, so that makes more samples, which would cost more money. But also we were researching college students, so keeping them for 6 hours in a room made it difficult, especially whenever they had to leave for classes sometimes. And also the population could have caused us to not get differences between treatments. So like I said, we looked at already healthy individuals that already had healthy levels of MDA, AOPP, glutathione. So there wasn't a lot of room for improvements potentially. So if we did more unfit populations where they naturally have different levels, it's possible that we could have gotten those changes. So for example, in one of the studies that I saw where astaxanthin caused improvements, they looked at pre-astaxanthin supplementation, and then 4 weeks later, they looked at just like their morning levels of what's called MDA. So one of the things we looked at, and they were able to improve their MDA, but the improvements that they saw they were still higher than our participants' pre-levels. So it is possible that our participants were just healthy enough to not get those changes from the astaxanthin supplementation. But with hydrogen peroxide, we did see significant attenuation with astaxanthin. So why do we think that happened? So I'm bringing this back down here. So this is a slide that are showing that how hydrogen peroxide is produced. You can see SOD and glutathione are still in here, but so here we have hydrogen peroxide. We believe that astaxanthin was able to either go in and reduce the hydrogen peroxide more quickly than glutathione could have on its own. So it could have acted as an antioxidant on itself. And it also could have prevented the hydrogen peroxide from just leaving the cell. So how I talked about in the beginning, how it can act as that net in the phospholipid bilayer, it could have potentially, even though there was enough hydrogen peroxide produced, it could have kept it from leaving the cell, because we were looking at hydrogen peroxide that was in the plasma, not necessarily in each cell alone. For the FatMax testing, we didn't see any change in oxidation rates or lactate levels or respiratory exchange ratio. So why do we think that we didn't see that? Well, it could also be the population again. So we did look at healthy individuals. There might not have been a lot of room for improvements, but also the previous research that showed the changes in oxidation rates, they measured it differently than we did. So they looked at more fit individuals, and they only looked at one intensity, versus ours, we were ramping it up every 3 minutes. They would just be at one constant intensity the entire time, comparing before and after supplementation. Also, some of the studies that they have done were done in rat studies. So the results from rat studies doesn't always translate over to human studies as well. And it could have potentially been the equipment we used too. So typically with fat max testing, you use Amoxis or Cosmed, which are more sensitive than the Parvomedics analyzers that we have. So potentially if we use a more sensitive analyzer, we could have gotten some changes, but obviously we wouldn't know that for sure unless research is done on it. Implications. Why do we care that we got the results that we got? How does this apply to everybody? So astaxanthin can be used to decrease postprandial oxidative stress, and most people spend most of their day in the postprandial state. Most people eat breakfast pretty early on, and so the rest of your day is in a postprandial state, so it's after eating again. And as Americans, we typically eat a lot of large meals. So for example, here on the side, we have a honey barbecue chicken strip sandwich from Whataburger. This one sandwich, so this, again, no fries, no ketchup, no drink added to it, 910 calories in this one sandwich. And I have yet to meet someone that's going to only eat the sandwich and absolutely no fries with it. And like I said earlier in our study, the average milkshake that we made was 1,169 calories. So this is pretty close to that amount, and you were able to see the changes in the plasma visually, as well as the MDA and AOPP actually increasing as well. And then down here, we have 10 wings from Pluckers. Doesn't matter what sauce you put on it. Again, no added ranch, no drink with it, no French fries. Anywhere from 945 to 1,531 kcals in that one sitting in that one meal. So this is something that's pretty regularly induced in most specifically Americans. And the American Council on Exercise, or ACE, estimates that Americans eat 3,000 calories on Thanksgiving alone, and specifically 226 grams of fat. So our milkshake on average was 80 grams, and this is over double that. It's almost 3 times as much fat as was in that milkshake. So this is postprandial oxidative stress is something that's very normal for everyday life for an everyday person. And additionally, even people that are pretty healthy, sometimes they adopt a eating protocol where they just eat one large meal in the entire day so that they don't eat consistently throughout the day. But that might not be beneficial because of the amount of oxidative stress that's induced. And why do we care if we induce the postprandial oxidative stress? Why do we care to try and decrease it? Again, going back to this slide that I had in the beginning, all of these things, so the high-fat meals, tobacco use, exercise, dysfunctional mitochondria, obesity, that lead to the oxidative stress can cause the heart disease and diabetes and decreased metabolic flexibility, impaired metabolism, tissue damage, and even lead to cancers. So we want to be able to decrease that oxidative stress so we can prevent all of those things from happening. And whenever you have a lot of these high-fat meals consistently, it can lead to all of this, even if you're typically a healthy person. So that's where astaxanthin could potentially come in, is it can go on any of these arrows and decrease the amount of oxidative stress. So it decreases the likelihood of these negative things happening to the body. So I'd like to go to the acknowledgements now. First of all, I'd like to thank my chair, Dr. McAllister, for really getting me involved in research and letting me do as much as I've been able to. I know I've been super lucky in the amount that I've been able to control with the research and the amount that I've been able to be involved, and for putting up with me on those early mornings whenever I was just really grouchy, or those late nights where we're just analyzing and just putting up with me in general. Dr. Mettler, I want to thank you for all the guidance that you've given me, as well as letting me get actually your research study that I helped with my first semester was the first time I was involved with research. Mr. Patek, we go way back. I took his class in spring 2016, and I started tutoring for him, and he's the one that kind of kicked off my passion for exercise physiology, really. So it's what led me here and led me to Dr. McAllister and Dr. Mettler to be able to do the awesome research that we do now. I want to thank Astaxanthin Incorporated for donating all of the supplements that we got for the study, so we didn't have to pay for that. I'd like to thank the participants for participating in the study, because if they didn't, I wouldn't have anything to present today. I'd also like to just thank all of my friends and family too. Everybody that's watching, I really appreciate all the support that y'all have given me. And I'd also really like to thank the researchers. So here's a picture that we all took together. So Justin, Dylan, Drew. Drew's the one that's put up with me the most, I think. And Cambry, Bailey, Mathis, Jessica, Shelby, all those super early mornings in the lab and making milkshakes at like 6 o'clock in the morning. I really appreciate all of it. And it was such an amazing experience. I feel like these pictures really capture us more as a lab group, though, where kind of just having fun, and it was a really great study and a really great opportunity. And it was really cool to be able to do my thesis as my first big study. So I really appreciate that, and I appreciate all of you. Yeah. Now we have questions.
Hey, Lily, this is Kent.
Hey.
I don't know if you can see me or not. Great job. Awesome presentation.
Thank you.
You're fortunate to work with some of the best in the business and our exercise physiology folks that they know what they're doing. So wow. But I did have just a couple of really kind of simple questions probably for you as well as your committee. One is when you did the cycle ergometers and you're doing the 50-watt, you're doing the 50-watt stages. How long did those stages last? Like how long did you have them at 50 watts and then go to 100 and then 150?
Each stage lasted 3 minutes.
3-minute stages. Okay. All right. And you went all the way out to 250 watts.
So we would go until the participants reached volitional exhaustion. So until they couldn't go anymore.
Okay. That was my question. Did most of them get to 200? Because I'm a cyclist, so 250 watts, that's pretty high output.
Yeah, most of them did not reach the 250 watts. We did have some, but most of them were just able to finish the first 4 stages, and that was pretty much it.
Yeah, wow, that's amazing. The second question, I've been fortunate enough to work with diabetic kids, especially back when I was back at Texas Tech. So we worked with kids who are obese, and so I'm kind of interested in the diabetes that goes along with that and everything. So with a diet, probably, I think you were recommending the Astaxanthin high diet. You're right. So I guess you're recommending a higher concentration of that in their diet, right? So what about exercise? Any recommendations, especially for those who are obese and or diabetic? Yeah, so how does exercise maybe offset that or play into that?
So with exercise, we naturally have inductions of antioxidants in the body, so just like acutely. But over time, as you exercise and train, you naturally have more antioxidants in the body. So with training and with exercise, you can get the same benefits as what astaxanthin would be doing with oxidative stress. So improve metabolic flexibility, improve functionality of the mitochondria in general.
Cool. All right. Awesome.
What's more powerful, Lily, training or supplementing?
Training.
I'm sorry, what was the question, Matt?
I was asking her, so what's more powerful or what's more recommended? What is more effective? Would it be chronic exercise training or taking a supplement like this?
Oh, yeah.
Exercise training for sure, but not all of America can be motivated to train. So at least supplementing of some kind can at least help a little bit with that. It's easier to tell someone, hey, take this supplement once a day versus, hey, exercise 30 minutes every single day.
Yeah, that was one of the problems we had with our adolescents, our diabetic and obese adolescents, was trying to get them to exercise. It's the motivation to do it and finding motivational ways to get them to engage in high intensity or even moderate intensity exercise.
Yeah, exactly.
Cool. Yeah.
And some helps better than no help.
Yeah, for sure. So great job.
Thank you. Anybody else?
Lily, I just wanted to say very nice presentation, very nice work here, very nice job. And I'll leave my questions for later, but I just wanted to make sure I said that.
Thank you. I appreciate it.
Hey, Lily, I have a question for you. Just for curiosity, just because since Kent brought this up, is there a lot of research showing that there's more effectiveness between or when eating foods that are high in astaxanthin versus taking a supplement of astaxanthin? In other words, is there research that shows that natural dietary consumption of astaxanthin is better or worse or about the same in regard to supplemented astaxanthin?
So from what I've read it is about the same whenever you eat enough of it. So with salmon, you can get astaxanthin in that way, but you have to eat more salmon basically. So to get the same amount that's in the supplements, it just takes more eating basically. But once you have the same amount, then they react similarly. So there's not really a difference whenever you do it like that. Another thing with astaxanthin is it is fat soluble. So as long as you consume it with fat. So our supplements had oil in it already, and that acted as the extra fat to where you could absorb it more easily. So if you did take a supplement that didn't have that added fat, it would be more beneficial to take it like with a meal or just eat the astaxanthin through salmon. But as long as you have that added fat whenever you consume it, they react the same.
Gotcha. Just one more follow up to that, just because Kent mentioned this as well. The foods that are high in astaxanthin appear to be foods that are typically not very economical, that are expensive. Is that fair to say?
Yes. That
astaxanthin, to get a decent amount of astaxanthin, the same as a supplement, it would be financially kind of hard because the foods that are high in astaxanthin are expensive.
Yes.
Is that accurate? Okay.
Yes. Yep. So I think one bottle of astaxanthin that I saw, it had like 60 capsules in it, I think was $15. So that's like the price of salmon, some of it. So buying the astaxanthin as a supplement is definitely more economical for sure.
Thanks. Great job.
Thank you. Any other questions?
I don't have a question, but I do want to thank the panel because your name's been spoken with high regard in our house for a few years now. And
It's
I
my mom.
appreciate it.
Thank you. Appreciate that.
She's been great to work with.
Thanks. Most days.
That much I knew. Yeah.
I do have one question.
Okay.
This is Jennifer. I'm Liliana's sister. So just to clarify, did I understand correctly that you gave the participants, the research participants, both A and B supplements, and they were supposed to alternate every day?
Not every day. So for the first 4 weeks, for the first 4 weeks that they took A, the second 4 weeks they would take B, or vice versa. So they would either take B for the first 4 weeks or A for the first 4 weeks, or the second 4 weeks.
Got it. Okay. All right. That answers my question.
Okay.
That was it.
I just want to say one more thing, and then I got to leave. I've got something else I got to attend to. But hats off to the committee, and especially Matt for directing this thesis. It was really nice work. Really nice work. Excellent. Thanks, Lily. Congrats. Good luck.
Thank you.
Well, if there's no other questions, Lily, great job. It was awesome working with you. We're not done yet, but in terms of working together. But if there are no other questions, any other questions come up?
So I just want to say none of the lab mates or friends want to ask a question to make Lily look extremely brilliant here.
I didn't see too many questions.
Y'all use?
They asked for questions, and I said I wasn't giving them any. They had to come up with them.
I think what we're going to do now is we'll break out, and people that aren't on the committee, if you'll please sign off, and we'll take time with the committee members and Lily, talk with her a little bit more about the project. Thanks everybody for signing on and joining. We appreciate that. We're not used to doing Zoom defenses, but we're glad that we're able to do it at all. So just making the best with what we've got. Appreciate you guys. Thanks for coming.
Yeah, thank y'all. Appreciate it. Happy birthday, Drew.
Thank you. Good job, Lily.
Thank you.